telling these kids about like goblins and like weird stuff like this, right? But this is kind of what we're doing right now, right? Which is that I have a bunch of patterns and sequences of brain activity in my head, right? That contains information about like what's in this talk, and pretty like complex information too, like pretty structured information about like the kind of work that I've done and about the modeling, the types of modeling that we do and things like that, right? And I'm going to take those kind of relatively like abstract, high-level structured brain representations, and I'm going to compress them down into a very kind of impoverished, simple serial transmission that I can send to you, right? Using language, essentially. And I'm truly going to basically, like, I'm broadcasting that to you through vibrations in the air, right? And because you have a similar kind of setup, right? And not only are you human, but you speak English, and we have a lot of similar cultural conventions and things like that. You're able to take that kind of packet, that transmission, and expand it back out into something resembling the brain states that I had in mind, right? And that's the kind of goal of what I basically want to try to reshape your brain activity to resemble, to some extent, my brain activity, in the hopes that in doing that, you will sort of understand what I'm trying to convey to you, right? Does that make sense? We can sort of, I mean, that's the kind of like very high level scientific framework that we work in, and that's not like a solved problem in any kind of way, right? So that's like the high level description, and then everything else we actually do probably sounds very unsatisfying relative to that, right? But so if we kind of, to try to like, dig a little bit deeper into this idea, right? So I'm going to zoom in on one of the listeners here, right? And try to sort of like figure out, okay, so we have this listener, right? And they're hearing the story, and we can actually use things like fMRI or ECOG in epilepsy patients, or EEG, or all sorts of various measurement tools to try to get like different views onto the brain activity that's happening in this listener's brain while they're hearing a story, for example, right? And if we do this, right, the first thing that the thing that's like most obvious, right, is that you have in superior temporal cortex, right, transverse temporal gyrus, you have brain areas that are kind of cortical systems that are like really closely tracking, the acoustic features of this incoming stimulus, right? They're doing this for music. They're doing this for, if someone were kind of just like, they're doing this for, ambient sounds outside, cars driving past and things like this. This area doesn't really, care too much about like what is going on, but it does a very kind of like rapid processing of the auditory input, right? Of course, it is very well tuned to kind of like human speech, right? Human speech is a very important part of our lives. It's one of the most important stimuli that we receive, right? So it is kind of like geared toward, extracting, phonemes from speech and like keying into particular intonation and sort of like rhythmic things that we do during speech, right? But again, relatively low level here. And I don't know if people, I mean, like this is sort of like even in your kind of ear, essentially, you have the cochlea is kind of essentially doing what is like a spectral decomposition of, the incoming sort of frequencies into different particular bands, frequency bands and things like this, right? And so that's the kind of like starting point. That's the entry point into this system, right? And as you sort of move from there, right, you start to accumulate information. So that's a very fast, that brain area, that blue brain area, only really cares about what was happening in the past, couple hundred milliseconds, maybe the past like second or two or something like that. But it's constantly sort of updating and bringing in new information, right? But as it's doing that, right, it's constantly feeding, whatever it gets that's sort of like interesting or mismatching or things like that. It starts feeding up into higher level systems, right? And so some of these it's not as strictly hierarchical as I'm kind of portraying it here, but one of these higher level systems is basically going to start resolving like syntactic and structural things, right? In language, things that I'm saying right now will kind of impact how you interpret, even just in a very low level grammatical way, the things that I'm going to say in a second or two, right? And things that I'm saying right now are kind of like being like certain ambiguities about what I'm saying right now are being resolved by things that I've already said, you know, in the past like second or two, right? Sort of even longer time scales as well, right? And so all of this kind of like, you know, the parts of speech and the syntactic kind of like dependencies and things like that are being resolved in sort of somewhat higher level. And a lot of these areas like this, I don't know if you guys can see my cursor here, right? Like this is also, you know, like even like frontal cortex, right? Even into kind of like motor areas, like we're probably using similar systems, probably co-opting similar systems to what we use for actually producing speech to kind of resolve some of these syntactic dependencies while we're listening, right? And then, you know, then at these longer time scales, right now you started to get into like the really interesting, at least in my perspective, really interesting stuff of like, how do we actually understand what people are saying? Of course, we sort of know what individual words mean, right? Like if I, you know, I can see a chimney out my window, right? That's a very completely out of context kind of thing. And you sort of know what that word means, right? But most of the words that I'm saying, the meaning is really highly context dependent, right? Like if you were to just present the words that I'm saying in isolation, right, they would not necessarily tell much of a story, right? So you have higher level brain areas that are kind of, again, an even sort of slower evolving kind of processing system further up the hierarchy that are trying to resolve these like long range or kind of almost like static contextual semantic properties, right? And then the highest level that we talk about in the way that we do research here is what I would call like narrative or event level processing, right? And this is happening over the course of, you know, it might switch in some cases over the course of seconds, but then we might sit in a particular situation or be in a particular event for, you know, many seconds or even minutes at a time, right? And that kind of long-term processing is very, I mean, that's a really difficult thing to kind of like latch onto experimentally, right? But this is something that's very important. Like we think of the world in terms of like I'm the speaker right now, and I'm sort of delivering a talk to you guys, right? But there's other people in the room, right? And we're in this particular context. I'm in my apartment and whatever, right? Those are all sort of factors in the kind of situation model that will then switch after this talk when I go to eat lunch or get coffee or something like that. So all of those factors are kind of like important in understanding what's going on here, right? So this is a very, you know, again, high level kind of view of what the language system is doing, right? And the idea here is that as you increase, you know, as you get further and further away from the input, you have more and more sort of input from like your pre-existing sort of machinery, right? And the kinds of like representations that the brain is working with in those contexts becomes more complex and more abstract. There's also, I think, underappreciated important kind of thing of like all of these brain areas are constantly working, and they have sort of priors built in, right? And the thing that's important is that the kind of information that they care about and represent it kind of evolves at different temporal scales, right? So low-level brain areas are really have rapid turnover, whereas high-level brain areas are kind of slowly fluctuating over time and pulling in information from, you know, seconds or even minutes or however long ago. Okay. Does that make sense? Anybody want to jump in with questions about just like this very like, you know, the very high altitude view of language in the brain? Again, this is not a solved problem. I'm not presenting like a mechanistic. I'm just sort of like pointing at parts of the brain and sort of suggesting what we think might be going on there based on that.
I had a question about feedback.
Mm-hmm.
Can feedback between, say, acoustic processing kind of eschew some of these higher level processing events, like like the feedback from the microphone where it's squeezed? Like, wouldn't that prevent semantic processing in a sense, or change event processing?
Yeah, that's totally true. So first of all, you definitely have, I think the way that we think about this as, first of all, this whole thing is sort of constantly cycling and everything is sort of talking to itself. It's not as strictly hierarchical as I was describing. I think the way to think about it, right, is that the higher level, slower areas are one framework for thinking about this is kind of the higher level, slower areas have sort of a model running, right, of what the kind of content that they deal with, right. And what the lower level brain areas are doing is like they're sending information up, and a lot of times that information probably fits with the model, but when that information doesn't fit with the sort of running model, that's a kind of like a prediction error or something like that. That triggers a kind of like change in processing, right? So it's like when unexpected things happen, or like you're saying, like if there's some something odd about like the incoming, like upward signal, right? That's what sort of triggers like the downstream systems to be like, okay, something changed here. I need to kind of switch my model or adjust my model or something like this. But yeah, there's constantly sort of like that kind of feedback, right? And it's not even really just like level to level. It's like even the higher level, there's probably sort of connections that skip as well, and all sorts of things like this, right?
So last question is, what determines the stability of these models that you are mentioning?
That's a good question, right? So you're talking about like the internal model of the environment that the brain is sort of like running, right?
Yeah, like I'm thinking about like the affinities that each of these has for how, like what it processes the information coming in off of.
Yeah, that's a really good question. I think I guess one way to think about it, right, is that all of these things are, we are like biological organisms created by evolution, and we are like the things that our brain is set up to deal with the things that are most like ecologically relevant for us, right? And so you have like these dedicated low level areas that have like a very fast sort of evolving, temporally evolving model, right? But we need to like, what one of the things that really like makes us human, right, is our ability to kind of like have very abstract high level representations, and those are probably like these slower evolving, temporally evolving models in higher level brain areas, right? So I think it's really just like evolution built us to have, built us to kind of have that kind of hierarchical multi-level model, and with certain parameters, like certain time constants or something like that, right? But I don't know if I have a better explanation as to like why it might be this or that kind of like number. Right?
Thank you.
That makes sense? Yeah, so that's kind of one direction here, right, in this process, right? And so I think the other interesting thing that we try to get at in like Uri's lab, for example, right, is there's a lot of other stuff going on here. First of all, like the speaker's brain is also doing a kind of like similar process, right? But the speakers also, it's not just verbal, they're using gesture, and they have facial expressions, and so you're getting like visual, multimodal kind of like visual input as well. There's like the people in the audience with you, like these other listeners, right? And if they start like oohing and eyeing and things like that, right, that will kind of impact how there's many social dynamics to how this thing works. And even just kind of like, okay, if we get into this, if we latch on to the speaker's brain as well, we can kind of see that we're doing a kind of like inverted thing here, right? Like the speaker is essentially like has high level abstract representations that they're trying to convey, right? And they have to compress them down. This is sort of what I was describing before. And the listener is doing this kind of opposite process of unpacking these representations that these sort of like tightly packaged linguistic representations that they're receiving. And the whole general framework that we talk about here with this is what I refer to as brain-to-brain coupling, or brain-to-brain like alignment, right? And again, this is like, again, the speaker here is trying to reshape your brain activity in a particular way to convey a particular idea to you. And that idea is just a configuration of your brain, right? I'm trying to recreate some configuration of my brain in your brain, right? And scientifically, this is a really tricky question to get at, right? Because first of all, most of the cool, aspects of human communication, especially language and things like that, are pretty uniquely human, right? So most animals do have some sort of communication, but it's difficult to study. You can't really study language in like mice or something like that. So a lot of our best like animal models and these incredible neuroscientific tools we have, like optogenetics and things like that, right? Very invasive kinds of tools, we can't really use to study language, right? So we're stuck with these non-invasive like brain imaging and things like that, which is often what we do in the lab. And language is also really, there's this problem with language that it's like, because of this kind of like temporally evolving dynamic structure that it has, and the fact that it's like fundamentally, meaning, for example, is fundamentally contextual, that makes it like really unamenable to our typical like, experimental design practices, right? So like we like to do things, you know, like psychologists love to do things like, okay, I'm going to control all of these variables except for this one thing that I care about, right? And I'm going to present these trials in like a randomized order, and I'm going to average across trials and things like this, right? None of those things really work with like natural language, right? Which is always, you're always hearing something kind of for the first time, and what you're hearing now really depends on what happens. So you can't sort of like shuffle those things around and do the normal experimental things that people would typically want to do, right? And so this is kind of, I would say, like the main innovation of Uri's group and the lab that I'm in, right, is moving toward more naturalistic experimental designs for neuroscience and psychology experiments, right? And that means that we're showing subjects movies, we're having them listen to like spoken stories, and we're you know, they're listening to their class lectures in the MRI scanner. Maybe they have an electrode array in at an epilepsy unit, but we're recording like what they're saying and things like that, so we can kind of compare that with the brain activity, and so forth, right? So trying to break out of this like classical psychological approach to experimental design and build a complementary approach that is more like ecologically valid, is like one of the terms that we would use. Okay, and that's very challenging. And so the last couple things that I'll talk about here are kind of things that I work on to try to make this easier, right? And it's a little more technical, but I'll still keep it pretty high level. So one of the like very core issues that we come up with here is like, how do we actually measure shared responses across subjects? And the simplest way to do that is this thing we refer to as intersubject correlation. And the way that you can think about that is, if we have sort of, you know, if I have a brain area here and you have the same brain area, right? And I'm speaking, or even if we're just both listening to the same sort of story or watching the same movie or something like that, we can measure the extent to which those areas fluctuate in time in a consistent and sort of reliable way across your and my brain, right? And if we look throughout the brain, right, at areas of our brains that are sort of fluctuating in synchrony, right, given a common stimulus, given like a shared stimulus, like a movie or a story or something like that, what that tells us is, okay, you know, you and I have different sort of machinery going on here, right? But there is enough commonality and there's enough sort of stimulus-driven signal that I can find brain areas that are basically responding in temporal synchrony like this, right? And so that's what the top row of this figure is trying to show here, right? Is I can basically take one brain variable and map it onto this roughly the same brain variable in your brain, correlate those time series, and I can get, you know, one correlation value that's basically like how synchronized are these subjects over time. Okay, or these brain areas over time across subjects. And, you know, if I do this between two listeners, it's like, okay, this is a stimulus-driven signal, right? I'm isolating the stimulus-driven signal here. If I do this across a speaker and a listener, you start to pick up more interesting things, right? Because the speaker is doing something different, but there are linguistic commonalities there. So, right, the sort of low-level properties, the low-level sort of processes going on between listening and speaking are quite different, but there are similar sort of high-level processes going on there. Another thing that we do, let's, you know, in this bottom row, right, is, okay, let's actually look at, you know, for every voxel in my brain, let's look at how the extent to which that fluctuates consistently with all of the voxels in your brain, right? And you might think, well, it doesn't make sense to really do that, right? It will, you know, the corresponding area of your brain is probably the same, but that's not, the brain is not really set up like that. The brain is set up in networks where you have a lot of sort of redundant kind of fluctuations and correlated fluctuations, right? So what this allows us to do is identify what we would refer to as like stimulus-driven like network configurations, right? And so this is what I would refer to as ISC, intersubject correlation, and ISFC, which is intersubject functional correlations or for connectivity. right, for identifying shared signals across brains and in time and shared network configurations across brains. We do this also in terms of like the spatial kind of response patterns as well. And this gives you a sort of different thing. Here I'm basically saying, you know, the fine-grained spatially distributed pattern of activity in your brain and my brain at a given time point, right? What's the similarity between those, right? And that might allow us to, that's a sort of punctate thing at a particular time point, but that might allow us to dig into like more sort of like multivariate complex representations of information in the brain than just looking at one brain variable kind of like fluctuating over time, right? This is just two sort of views, the sort of two simplest kind of views onto measuring brain-to-brain coupling, right? Which is in time, fluctuations over time, and in space, spatially distributed response patterns. And we do that in other things as well. The next thing that I'll just mention here briefly, this looks like very complex, right? But is that it's not actually as straightforward as I was just making it sound, right? Like, what are the corresponding brain areas between yours and my brain, right? And, you know, given the fact that not only are our brains like differently shaped, right? We can sort of use image registration algorithms, and we can sort of align this, or at least like rough shape of our brains, right? But even if I were to perfectly align our brains, it's not necessarily the case that like the actual functional sort of properties of a given chunk of brain tissue between your brain and my brain are that similar, right? They might be sort of, you might have similar functionality, but it might just be displaced by a millimeter or even a centimeter or something like that. And that's just because we, you know, all develop differently. We have different experiences, you know, throughout development, right? And so the mapping between function and anatomy is it's roughly consistent, but at a fine grain level, it's not that consistent across people. And so another one of the things that we work on is this is what I would refer to as like a hyperalignment tool. This is a functional alignment algorithm, right? And the way that we think about this is I can take all of your brains and kind of treat this as like a matrix sort of decomposition problem, where what I'm going to try to find is some I'm going to allow, you know, I'm going to allow for some kind of like shifting around of functional responses. I'm going to sort of try to rotate your brain space to line up the functional responses with my brain space, right? And what that's going to give us is, first of all, a shared functional space, right? And also a set of transformation matrices for taking any given person and kind of rotating their sort of brain features to kind of line them up with the functional activity in another brain, right? And what this allows us to do is get much finer grained, sort of much more functionally precise alignment of brain. So if we were to do, you know, these ISC analyses that I had previously described, after doing this, we get a lot more signal out of it. We get a lot higher dimensional kind of like shared representation if we use these more sophisticated alignment algorithms. And the last thing that I'll mention here is that we just, to better understand these things, we need like more data, right? Like one of the things that's really hot right now is computer science people, machine learning people are creating these language models like GPT-2 and GPT-3 and BERT and XLNet and things like this that can do I mean, they're trained on billions. They have billions of parameters, and they're trained on billions of like text tokens from the internet, right? But they can do incredible feats of like, they can do incredible language things like summarization and basically like kind of fluent conversational stuff. Of course, they still get a lot of things wrong and they don't really fully understand what's going on, right? But we want to be able to start leveraging these new computational language models from the NLP community, right? But to do that, we need more data, and we need like big sort of like benchmark open data sets to kind of like accelerate research in that kind of way, right? If you think of like things like ImageNet and the way that ImageNet kind of accelerated deep learning, we want to try to create similar things for the neuroscience of language, right? So one of the main things that I work on is kind of curating and sharing big data sets, right? So this is our narratives data set, is a data set like this, right? And the point of this is that language natural language, like a spoken story, right, has so many things nested into it that you can test many, many, many hypotheses against it, right? Like you can, if you care about the acoustic features or the semantic features, the narrative features, or if you have, you know, all these different sort of models, we can compare all of them against like a rich enough data set. And that's the thing that we're trying to compile here with narrative. So this is a data set where this is an archival data set that I put together of 300 plus subjects, almost 900 functional scans, which amounts to, we have 27 different stories in this data set. It's about 4.5 hours of unique stimuli, and it comes out to about 6 days worth of fMRI data if you were to kind of stack it all up like this, right? And we put a lot of effort into, you know, providing transcripts along with this and standardizing the data for automated processing and creating all of these derivatives that are sort of ready to use. So, you know, NLP people don't love pre-processing fMRI data, so we try to give them the kind of like final product so they can run their models up against that. And just to give you an actual kind of brain image here, this is the first thing that I was talking about, intersubject correlation analysis applied to this pretty big, probably the biggest natural language imaging data set out there. And you can see, right, that first of all, across all data sets, right, you get highly consistent responses throughout a large swath of cortical tissue. And all of these, so it's very similar across different stories here, right, which I think is encouraging, right? It's very consistent across the different story stimuli. And what's most important here is like, these are reliable signals across subjects, but they are all different, right? So, you know, these early brain areas, right, are highly reliable. They're really closely sort of yoked to the auditory stimulus, right? But even these sort of like more lateral temporal areas are doing are processing much different information. It is just also sort of stimulus related and reliable across subjects, right? So at every single one of these brain areas, we can start comparing different models of what's going on, right? And that's the kind of like, to try to scale this up to like a larger scale kind of community-oriented science. These are the kinds of resources that we need. Okay, so I see I'm running out of time here, so I think that's pretty much the end for me. I want to thank all the people in Hasson Lab and our funding sources, and thank you guys for listening.
Hey. Thank you, Sam.
Thanks, Sam. Thanks so much.
Thank you.
Just going to open up to any questions if anyone has any. Oh, okay. We have one from Amar. Oh, we have a lot. Amar, would you like to read your own question?
Yep.
Okay. Okay. Well, we're waiting for Amar. Sebastian, go ahead and ask a question.
Thanks for this interesting talk, Sam. Could you go back one slide just to ask you something to see if I really understood what? So all of those brains on the right of the slide are like showing the areas with the highest intersubject correlation for a particular story?
Yeah, yeah.
So why some stories like are more or trigger more correlation, I don't know how to say it, in the brains than others? For example, I see that the 21st... Sorry.
Yeah, I think in a lot of cases, it's not necessarily for like interesting reasons. I think the thing to keep in mind here is even though all of these maps look the same, right? All of these stories are completely different, right? There are different durations, different content, different speakers, and things like that, right? And so all we're measuring here is kind of within that story, how reliable is the signal across subjects, right? And that is kind of agnostic to the content of the signal, right? So that's why the maps look similar, even though there's all sorts of different stuff going on. It is totally true that some of the maps aren't that perfectly similar, right? And I think the reason for that is like, you know, some of these stories are like 21st Year is like almost an hour-long story, right? So that's just a lot more data going into that, whereas something like PyMan is like 8 minutes long. There's also boring things like PyMan's a lot older and it was collected on, using an older scanner with older kind of parameter settings and things like that, right? So I've tried to kind of harmonize all of these data sets as much as possible here and kind of, equalize as many things as possible, right? But there's just a lot of differences going on. There are also different samples of subjects, right? That, PyMan has like something like 90 subjects, whereas 21st Year has something like 25 subjects or so, right? And those are largely non-overlapping pools of subjects, right? So there's a lot of, there's a lot of stuff going on, right? We try to think optimistically about that as like, you know, more opportunities for modeling, but it's you know, with archival data like this, it's always kind of a mess like that. So
I see. Thank you.
other questions?
Amma,
Go ahead. Yeah.
Amma has a comment. Their bandwidth is very low, so they cannot turn on their mic. I will read their comment. As human beings evolve through the years, the only one thing that does not change over the million of years is conveying message through storytelling. Storytelling is coded in our blood, and that's why we connect with film storytellers and novels. And then they say there are a bunch of chemicals that make people laugh, make people happy. Sam, have comments?
I think I think it's totally true that I think it's hard to underestimate the importance of things like communication and storytelling in like human evolution, right? And the way that I think about it is not even like it's obviously biologically important on like an evolutionary scale, right? But we do this really weird thing, like like, you know, like other organisms do like niche construction and things like that, like you know, a beaver will build a beaver dam, or like, you know, other organisms do like weird things to their environment to kind of help themselves out, right? But humans do this crazy thing of like, first of all, telling stories, and also just like writing things down, right? In this way that accumulates a kind of like cultural knowledge. And so when we think about like, what does a human brain like learn, right? I think it's really crazy to like, you have to basically take into account the fact that we have accumulated a ton of cultural knowledge, and what I'm learning is like, I'm reading from books that are like 100 years old, and I'm hearing from people who have heard it from somebody else and things like that, right? You can't really consider a human brain in like complete isolation, right? Like if you raise a human in complete isolation away from like that's not an ethical thing to do, obviously, right? But it's like, that's like, you know, that person, that kind of the brain of that kind of brain will be like fundamentally, you know, disabled, right? Like it's not like that's the kind of the level of importance of like being immersed in the social context that we're immersed in, and talking with people, and also absorbing all of this kind of information that's encoded, that we, as you know, previous generations of humans have encoded in the environment, is, yeah, it's hard to underestimate the importance of that, for sure. And and like things like chemical signaling, right? Like, you know, this is MRI for the most part, right? We don't even have access to any of that cool stuff, right? It's really hard to, first of all, it's really hard to like, we can sort of look at that in kind of a static way, right? But it's really hard to see like the rapid temporal sort of progression of chemical events that are happening, you know, when like the story really picks up, or the movie gets scary, or these kinds of things, right? We just don't have access to that, unfortunately.
So, do you want to go, Penelope? Sorry.
No, go ahead, please.
Okay. So I had a question about as far as the way information is conveyed and learned. Have you thought about how some of the brain morphology, or changes in brain morphology that you showed before, might correlate with learning style, as in, you know, physical, what are the other ones? Physical, visual, kinetic, etc.
Yeah, I haven't worked on that kind of problem. There are people that are working on this kind of thing. And so I don't know if I have a very I don't think I have anything really very sophisticated to say about it, right? But one of the things that we often, like a very sort of simple result that we kind of observe a lot, right, is just like, if I scan 30 people who are listening, and we have some studies doing this. If I scan 30 people that are listening to like, okay, I take a teacher, like my colleague Mai, right? She knows a lot about fMRI. We put her in the scanner, and we had her basically like teach a lecture on fMRI while scanning her brain, right? And then we have 30 other people, students, that are going to come in and sort of listen. They're going to go in the scanner one at a time, and they're going to listen to her lecture, right? you definitely observe this effect of like the people looking at the variability across that population of that sample of listeners, right? You do observe this thing of like the people who end up aligning better with one particular person are often the people that will end up having like our measures of brain alignment are higher in people who will then, you know, score higher on like a test about that kind of material, right? So there's, but it's really hard to like systematically kind of like look into this, right? Because you would probably need like different teaching styles and you know, and things like this to really get into that kind of. But I think that there are groups out there. I'm thinking of like Dave Kramer from Dartmouth, for example, right, and Josh Cetron, who's at Harvard now, and who try to go in and like, you know, look at scan like first year physics students or something like that while they're learning like fun basic physics concepts, and then compare them to like the brain representations in the lecture or things like that, right? So like looking at expertise and looking at how different learning styles might contribute to that. So I think people are doing that work. I just don't really know I don't have much to say about it.
Okay, and then can you go back to the slide where you showed the distribution of participants? You had like 304 reported as female. Yes, okay, 204, thank you. So I was wondering, do you, with regards to like the way that the story is told and who is telling the story, I was wondering if you are worried at all about like cultural representation, representing, you know, different minority groups within the data set and how that might, you know, change the way that stories are told and the way that different parts of the brain are hit.
Yeah, I think that's a really important thing, and I think we certainly have not paid very much close attention to it. In like, again, this is some of these data sets are 7 or 8 years old, right? We typically try not to like exclude too many sort of like there some, you know, studies, right? It's like, okay, you have to be right-handed, you have to be a native English speaker, you have to be between these age groups. We try to get like a pretty broad sampling of you know, as broad as we can of people, right? And again, we just track all of that demographic information, so if somebody wants to model it, they can, right? But that said, these are like Princeton students and Princeton sort of, you know, adjacent people and things like that, right? So it's not a very kind of diverse sample, and it probably makes a huge difference. I think that there are more recent larger scale studies that try to take this kind of thing into account. There's like, one of them is like the adolescent brain cognitive development ABCD study, you know, where they're scanning, I don't know, like 10,000 kids or something like that every year over the course of adolescence. They're starting at like age 8 or 10 or something like that and keep scanning them, right? And so those groups, I think, have done a better job of developing protocols for kind of like for not basically like creating a very biased sample, for having a sort of diverse sample with enough sort of variability that we can model these things, and so that we're not just talking, you know, you've heard this idea of like weird brains, like white English speaking whatever kind of brains. And I think the other really important thing to keep in mind there, and I think some groups are starting to get into this too, is just like when you're doing really large scale, like tax funded studies like that, or consortia style, like data collection efforts, right? It should not only be, like when we're thinking about like, okay, what kind of, you know, consent do we need to get from people and things like that, right? I think it's important to also bring just, you know, kind of like broad people, like a broad slice of the community into the kind of decision-making process as well, right? So it's not just that we're going to try to collect like a diverse sample of subjects, it's that we're going to try to build a sort of community-based committee to guide like the actual design of the study and make sure that we're not just sort of like missing things, right? Because it's totally the case that if you were to put all of the, you know, PIs that are designing that study into a room, most of them are older, most of them are white, most of them are men, and so forth, right? And so trying to like create these more community, you know, like steering committee kinds of things, right, I think is another really important aspect of that.
Okay, so I have just a silly question that, oh, sorry.
A little running out of time, so yeah. And send Sam emails or
Yeah.
post Neuro.
If you guys have other questions, send me an email too, for sure.
Thank you so much. Thank you, Sam.
Sam, thanks so much for joining us. That was a really beautiful talk to show us how, if you can get, you can actually change the activity in someone else's brain depending on how you convey information. This brain-to-brain coupling is
what we're doing all the time, right? That's the only way that it could possibly work. So
yeah. So let's tell some stories then. We have a few minutes at the end here. We just want to introduce you to some resources. And I'm going to share my screen with our mural. And you know, we started thinking, Hannah and I, about the idea of preparing you for ways to convey your stories, your science stories. And we're using as a model that Princeton Research Day, that 3-minute video. I know some of you will not participate in that, but we're thinking like, let's use that as a potential project. And if, you know, Sam has now convinced us that the way that you communicate, and if you get past sort of just syntax and semantics and get to conveying an event, you can really tap into someone's thinking a little bit more. So we want you to think about, and we posted these resources here. One of them is this science storytelling workshop by Sarah El-Shafi, and she has a really a great workshop posted that you can access, taking you through the steps of how can you tell a story. And the first step is to connect with your science, and then to connect with your audience, then to craft your story, make characters, even if it's sort of a stretch, you know, maybe your character is some sort of cell type or a molecule, and then set that story in motion. And then the takeaway for your audience may be much stronger. So we invite you to look at these resources. One is, again, that workshop, and another one is this Storytelling Academy that Pixar in a Box presents. So we were saying breakout rooms, but I think that there's, well, yeah, I think we will put you in breakout rooms. What we wanted you to do was start to assemble a story in this format. Once upon a time there was, every day, one day, because of that, until finally. And I just gave a sample, you know, my research, and it actually, as I was putting it together, it's not too story-like. It's still kind of not event-based enough. So this would need to be revised, but it was sort of like, once upon a time, there was spinal cord injury, you know, 20,000 mostly males a year get spinal cord injuries. And every day, where's my format? Every day, neurons tried to regrow across the spinal cord injury, but these glial cells were really stopping them. Until one day, we realized that neurons grow really well on soft materials, but glial cells don't. So what does that tell us? What is it? Because of that, maybe we can create biopolymers that are soft enough that neurons will regrow on them, glial cells will not, and the neurons can then span across the transection of the spinal cord injury until finally, perhaps we'll have working circuitry again. So I would like you guys to approach your work in pairs to say, can I set up a story that way? And of course, we want to be careful. Science storytelling, there is some critique about it that we've found in the literature, that maybe it's too persuasive one way or the other, or maybe it cuts out important information. So you can keep that in the back of your heads. But let's see if we can frame some stories for the next few minutes. And then this may be something that we continue next time, or maybe we have time to come back for one example.